Our next presentation is STRM Bio, and I'd like to welcome Michael Luther, CEO and Board of Directors, to the stage.
Good afternoon. Today I want to talk to you about STRM Bio. We're a seed stage company that is focused on providing the next generation of delivery platforms to overcome the current hurdles that exist in the world of cell and gene therapy today. As we know, today, if you think about AAV and LNP, there's non-specific tropism. There's the issues that they're not allogenic, and so you're limited to one dose. You're also limited by the size of the cargo, and obviously there's the issues we have with manufacturing and scale up in terms of going forward. What we have focused on is a natural approach using extracellular vesicles produced from megakaryocytes that we can then load with multiple payloads. And the fact is that these extracellular vesicles are allogenic. They have natural tropism to human bone marrow, and they're not limited in terms of cargo size. To date, we have not found anything that we could not load, and we're still pushing the boundaries of that as we go forward today. In the last year, we've made tremendous progress in terms of the activities that we've regarding this platform that sets us well to actually both deliver on a platform and move forward on a pipeline. We've developed a consistent process that allows us to scale. We've been able to show that we can load it with diverse cargo, and I'll go through some data showing you we can do both protein, DNA, and RNA. Most of our experience to date is in RNA, and I'll go through that. Secondly, we've been able to show cargo-specific expression in bone marrow, both in multiple models, and I'll go through some of that data. We have tolerability. For example, in NHPs, we can show both biodistribution to the bone marrow in NHPs, but also we can show that we can dose up to 4 times without any safety signals to date. And that is only limited by what we've actually been able to dose so far. And in the last is that the fact is that we've now set ourselves up to actually start making that turn in terms of moving towards being a pipeline as well as platform-based entity in terms of both disease modification and moving forward in terms of showing that we can actually express a specific protein in NHPs that is related to an efficacy, moving from a process that is now around 25 liters to going up to 50, 100, and 200 liters that will allow us to actually be in a good position for IND enabling studies, and then able to actually show first in human profile. In terms of our process, we actually use blood, and from that blood, we actually isolate peripheral stem cells that we can then differentiate into megakaryocytes. And using a unique process in terms of this differentiation, we can then purify and isolate these megakaryocyte extracellular vesicles. Now, these are not just exosomes. These are extracellular vesicles in the 100 to 300 nanomolar distribution range, but in those extracellular microvesicles from megakaryocytes, they have natural tropism, while exosomes themselves may not. And what you can see here in this slide is just the isolation of the vesicles. By the way, once we isolate these vesicles, we can freeze them. They're stable. And we can show that actually in terms of consistency, in terms of growth, biomarker profiling using various proteomic-based approaches, in terms of yield, and then in size that we see commonality. We load these using a polymer-based approach. The polymer-based approach actually loads the payload on the outside of the vesicle, as shown in this electron micrograph here, and we can show we consistently can do that and deliver it in vivo. As I said earlier, that we can do this both in mRNA, in plasmid DNA, as well as protein, and we can show that we can do it in multiple doses, and that at the end of the day, we have a bankable product which we can then take and test for various activities. Here just shows a bone marrow-specific protein expression in which we've taken luciferase-labeled mRNA and then shown then on the left here, you'd see LNPs that have been labeled, and on the right is our megakaryocytes labeled vesicles. And you can see that while there's a lot of expression in liver for the LNPs, the only place we actually see labeling is actually in bone marrow. More importantly, while we see, because this is a non-perfused model, we do see some of these vesicles in liver, but the payload itself is only expressed in the bone marrow. And this is something we can see consistently, whether we do this in that luciferase model, or if we move to a more Crelox type model where we can show both a dose response here in terms of whole bone marrow. You can see multiple doses, different doses that with lower doses we see greater sensitivity. And not only are we seeing delivery to whole bone marrow, we're actually seeing it delivered to the specific types of cells that would lead to an efficacious effect in terms of going forward in terms of our pipeline. This is I skipped one, but this is the Cre-loaded microvesicles, which is a very, very sensitive model. All you need is one combination event to actually see expression. And again, we see in this model only labeling and expression of the payload in whole bone marrow, compared to that of LNPs, where you see similar expression in whole bone marrow, but you can see you almost see 100 times more expression in liver versus these megakaryocyte extracellular vesicles. We've also done biodistribution studies in NHPs, and this shows the results where we've done IV. By the way, these are all IV delivered. We don't have to go directly into bone marrow for the labeling. We give these both by IV. And you can see here, shows both, we've labeled the vesicle, which is in the orange here, and then we've put, this is not protein expression, but just a marker showing labeling of the DNA, and it shows that the vesicle and the DNA are located and expressed in the bone marrow. Again, this is a non-perfused model, so you can see we do see some labeling in other cellular tissues. Now, while we don't have the data here, what we've been able to show is that we've been able to dose in NHPs 4 times and not see any types of cytokine storms, liver damage, or other types of toxicities that one would potentially might expect. Our pipeline is today focused on 3 disease areas. Fanconi anemia, and that's more historically because of the great relationship we have with the Fanconi Anemia Research Foundation, and this was really around gene replacement. Secondly is in gene editing, and that's around sickle cell disease, and we have some proof of concept experiments going on in vivo currently. That'll be a next step forward. And then last is really around gene expression, and through a recent funded grant, we'll be able to pursue in vivo CAR T with a number of different collaborators. And this is where really what opens up to us is we have this strong platform that enables to actually provide a naturally occurring extracellular vesicle, which is allogenic, it's not limited by size, and it allows us then to load it with these specific payloads. We realize what our expertise really is around in the platform side, and we have the ability to actually move forward on our own in terms of pipeline, but we're also looking through partners and collaborations, and obviously we're actively outraising monies as we want to move towards the next area. Our focus, because of the natural tropism, is hematological diseases. And it is a very large market and a large opportunity. And you can think of diseases like beta thalassemia, you can think of Gaucher's, you can think of sickle cell, Fanconi, which we've already mentioned, and then we've also mentioned other myeloproliferative approaches and diseases that we can go forward as well. And so, we feel like we've really started to turn the corner in terms of our proof of platform and setting ourselves up well for the future. And we're looking here in terms of this meeting is actually, can we find, partners, collaborators to help us move forward as we go to the next step. And last, I just want to thank the team. A lot of this, we've had strong support for both from our current investors as well as a number of different grants from NIH, Gates, and others that allows us to go forward. And I'll with that stop and take any questions. Thank you.